Hello everyone. Today we continue talking about the ear. Having dealt with the anatomical part, today we discuss the auditory process and auditory pathways. The hearing is the process by which sound vibrations transform from the external environment into action potentials. The ear is equipped to distinguish different characteristics of sound, such as the frequency and the intensity of sound waves. Frequency measurement is in hertz, cycles per second. The human ear can detect frequencies between 1,000 and 4,000 hertz, but a young ear can hear frequencies in the range of 20 to 20,000 hertz. Cats reach frequencies 3 times higher than ours. Elephants can detect sounds between 16 hertz and 12,000 hertz. The wax moth is able to perceive higher frequencies than any other animal. In fact, it can record sounds in a frequency range of 20 to 300 kilohertz. The intensity of sound is measured in decibels. The range of human hearing on a decibel scale is from 0 to 130 decibel, where the sound becomes painful. All these physical properties must undergo transformations to enter the central nervous system. Sounds are then initially picked up from the external environment by the auricle. The sound waves then travel within the external auditory meatus, whose purpose is to protect the tympanic membrane. The first transformation consists of the conversion of sound waves, the air vibrations, into tympanic membrane vibrations. These vibrations then get transmitted into the middle ear and the ossicles. The middle ear acts as mechanical transformers and amplifiers so that the sound waves reach the organ of Corti. Their amplitude is 22 times greater than that of the air impinging on the pinna. Without the conversion of sound energy into mechanical energy, the energy reaching the inner ear would be less than the energy reaching the tympanic membrane. The ossicles amplify sound in 2 ways. Firstly, because they are articulated to act as a lever system. The malleus is arranged so that one end is attached to the tympanic membrane, while the other end forms a hinge with the incus. The opposite end of the incus is fused with the stapes so that they act as one bone. The footplate of the stapes is loosely attached to the oval window of the cochlea, allowing it to move in and out like a piston. Second, the ossicles concentrate energy from a large surface, the tympanic membrane, to a smaller one, the oval window. In fact, the pressure of the sound wave increases in proportion to the ratio between the 2 areas, considering that the area of the tympanic membrane is about 55 square millimeter and that of the oval window 3.2 square millimeter. The pressure on the oval window is about 17 times greater than that of the tympanum. Pressure is defined as the force divided by the area perpendicular to the force over which the force is applied. The pressure in the middle ear cavity is ensured by the periodic opening and closing of the Eustachian tube. If the pressure in the middle ear were different from that in the ear canal, this would result in inefficient sound transmission. With equalized air pressure, on the other hand, the eardrum can vibrate properly and transmit sound. The stapes makes contact with the oval window with a piston movement that transmits vibrations to the perilymph inside the cochlea, whose upper part, the vestibular scale, communicates with the middle ear through the oval window, and whose lower part, or tympanic scale, communicates with the eardrum case through the round window. The cochlea can be considered as a rigid-walled canal rolled up on itself. But if we unroll it in a straight tube, we understand how, being incompressible, it is able to transmit the pressure increase along the scala vestibuli, where it meet an elastic and deformable part, the basilar membrane, which consequently curves downwards. This in turn causes an increase in pressure in the scala tympani, and, therefore, an eversion of the round window membrane, which is associated with an intraflexion of the secondary tympanic membrane, also called round window membrane. Tonotopy, also called frequency-to-place mapping, is an important feature of the processing of sounds by the mammalian auditory system. At the level of the basilar membrane in the cochlea, a tonotopic arrangement implies that high-frequency stimuli evoke largest displacements at the base, near the ossicles, and low-frequency sounds have their greatest effects at the apex. At the entrance of the cochlea, the base, the basilar membrane, is thick and stiff. Towards the tip of the basilar membrane, it becomes thinner and more flexible. A specific frequency of vibration thus preferentially resonates at a specific distance from the base. The cochlear canals contain two types of fluid, perilymph and endolymph. The perilymph has an ionic composition very similar to that of cerebrospinal fluid, which is similar to the composition of extracellular fluid. The location of perilymph is between the outer wall of the membranous labyrinth and a wall of the bony labyrinth, filling the scala tympani and scala vestibuli. The perilymph contains a large number of proteins, such as extracellular enzymes and immunoglobulins. It has a high sodium concentration and low concentration of calcium and potassium. Endolymph, also known as Scarpa's fluid, is a clear fluid found exclusively in the scala media that is within the cochlear duct. It has a unique composition not present elsewhere in the body, characterized by a high concentration of potassium, which helps maintain the endocochlear potential, and low concentrations of sodium and calcium. The sensory organ of the cochlea is the organ of Corti. As described in the anatomy part, the organ of Corti contains 3 rows of outer hair cells and 1 row of inner hair cells, surrounded by supporting cells. These are located on the basilar membrane. When the oval window moves, waves are transmitted through the perilymph fluid within the scala tympani and then into the scala vestibuli of the cochlea. As the fluid moves through these compartments, the basilar membrane shifts relative to the tectorial membrane. Acoustic information is primarily transmitted by the electrical signals generated by the inner hair cells, whereas the main function of the outer hair cells is to amplify the incoming stimulus. The detection of a sound stimulus and its conversion into an equivalent electrical waveform, a process known as mechanoelectrical transduction, occurs when sound-induced movement of the basilar membrane excites the hair cells by deflecting their stereocilia. Displacement of the hair bundle toward the tallest stereocilia leads to the opening of mechanoelectrical transducer channels, located at the tips of the shorter and middle rows of stereocilia. Deflection in the opposite direction reduces the likelihood of channel opening. When these cation channels open in the hair cells, potassium ions flow into the cells, causing depolarization. This depolarization leads to the opening of voltage-gated calcium channels. The influx of calcium triggers the release of neurotransmitters from the hair cells onto the auditory nerve fibers. This activity generates an action potential that propagates along the cochlear nerve and through the auditory pathways, ultimately reaching the cochlear nuclei in the brainstem. That's all for today. In the next video, we will discuss hearing loss in 2 diagnostic tests, the Rina test and the Leber test.